Hello, everyone. Welcome to the TIDES Digital Week, brought to you by the organizers of the TIDES conferences. My name is Michael Keenan. I'll be your host for today's session, which is titled Critical Attributes of mRNA Manufacturing for Early Phase Therapeutics Development. First, I'll cover some quick housekeeping items. Number one, if you experience any difficulties with audio or advancing slides, please refresh your screen using the F5 key. That's F like Frank, number 5. If you are experiencing any other issues, hit the question mark button to receive assistance. At any time during the presentation, you can submit your questions into the Q&A window on the left-hand side of your screen. In 24 hours, you'll receive a link to watch the recording of this session, and you can also download some other information on the right-hand side of your screen. Let's now begin by introducing our speaker from GenScript USA, Dr. Fengmei P., Head of RNA Biology Department. Thank you for joining us today, Fengmei. Now I'd like to hand it over to you to begin the presentation.
Thank you very much for the introduction, Michael. Hi, good morning, everyone. It's my great honor to discuss with you about the critical quality attributes of mRNA manufacturing for early phase therapeutics development. So RNA-based therapeutics are becoming the third milestone in drug development history, following the chemical drugs and protein-based drugs. The success of mRNA vaccine against COVID led the way to mRNA as a new drug class. The advantages of mRNA as a therapeutics have the mRNA don't integrate into genome, so there is a low risk of the insertional mutagenesis, and mRNA are only transiently active that can be metabolized, and the production of the mRNA can be a platform technology that's relatively simple, and it doesn't need to enter into the nucleus to be functional. So how does mRNA function? Let's have a quick review of this together. I believe we are all very familiar. So the mRNA includes a 5' cap, a 5' UTR, open reading frame, 3' UTR, or polyA sequences usually. Like the 5' cap is important in eukaryotic cells to bind to the translation initiation factor to initiate the cap-dependent translation. It's very crucial for the mRNA translation efficiency. The 3' UTR serves as a hub for post-transcriptional coordination as targets for mRNA and protein binding sequences. It's very important for the mRNA stability, and it also directs mRNA to translocate into subcellular regions. Poly-A tail makes the mRNA to be more stable, and it prevents it from degradation. Of course, the codon is the coding sequence. It can be also optimized to improve the translation efficiency. We'll talk about more about this later. So yeah, actually we have the platform to make mRNA for our customers. So during the mRNA manufacturing process involved these 4 key steps. So first step is start from plasmid production and linearization. Secondly, the in vitro transcription reaction. Thirdly, the purification. It can be simply purification, or it can be, or if at a more advanced requirements, it can be purified based on its poly-A tail using oligo-DT column, or we can perform further purification to remove double-strand RNA to make it more high quality for clinical trials. And finally, there is a QC step for the mRNA production process. Yeah, I want to mainly discuss with you today about the critical quality attributes of mRNA manufacturing. Here I list the 6 main areas. So first is the quality of the DNA templates. Secondly, the integrity and property of the mRNA itself. Then there are some key areas that I want to discuss with you today about the polyA tail quality, the capping efficiency, and the chemical modifications, and finally, the impurities of the mRNA. So first, look at the DNA quality. The DNA template quality includes considerations for its codon optimization, the integrity of plasmid DNA, the polyA tail quality in the DNA template, and the impurities in DNA. So for the DNA template, firstly, the DNA quality itself, when we look at that, the ratio of leaked original plasmid is very important. Since the leaked plasmid urinates a sign of there is that will cause the aborted transcription for the mRNA production step. So for that, we would like try to achieve highest unleaked ratio as possible. So that will be reflected as the supercoiled DNA plasmid ratio in the plasmid DNA product. Sometimes including a long polyA tail can reduce its supercoil ratio. In polyA tail verification, that's also very important. So the polyA tail length is directly translated into the mRNA polyA tail length and quality. And also longer polyA could help with the mRNA expression efficiency. About the impurities, actually there are a lot of them, so I would like to discuss about a key one, that's the host cell DNA residue. So the genomic DNA contaminants can impact the transcribed mRNA integrity. We did some study showing like less than 5% is more preferred for the mRNA production. So talking about the codon optimization, there are multiple things we can optimize in the codon sequence to improve the mRNA performance. So like delete or mutate the destabilizing motif or the mRNA cis element motif or the RNA's recognition sites. And we can also optimize the GC content and to optimize the codon to have a higher tRNA utilization. So with that, the GeneSmart software from GeneScript has a very good, have been used by researchers for the mRNA sequence optimization and showed good results. I would recommend to use that if you would like to. Yeah, about the quality itself for the DNA plasmid products, we would look at the plasmid supercoiled ratio. On the left side show you HPLC results. So there are 3 peaks that you can see that can distinguish of the open circular DNA, the supercoiled DNA, or leaked DNA. So in most cases, we would like to keep the highest ratio of the supercoiled DNA plasmid population. And we also talked about the impurities that could impact the DNA quality and mRNA performance. So the host cell DNA residue is one, as one example I want to share with you that we analyzed the ratio of different host cell DNA impacts on the mRNA translational efficiency. We can see less than 5% of the host cell DNA residue can generate a more stable mRNA quality. The second quality is about the integrity of the mRNA. So the integrity of mRNA, we can explain simply by the length, integrity by length, and that will significantly impact the mRNA expression. The integrity of the mRNA length and its expression coordination we have studied for 2 mRNAs in our hands, like the EGLP mRNA and the ESP-Cas9 mRNA. So here we compared some, for EGLP mRNA, we compared several products that we studied. We can see the higher integrity will translate directly to higher transcription efficiency. expression efficiency in cells. Same as the ESP-Cas9 mRNA, so those are exactly same sequence, just with slightly difference in the quality of the integrity. We can see this also directly impact with the mRNA expression efficiency. The third quality attributes, we would like to talk about the polyA tail quality. So polyA tail quality include its length, the polyA tail, the distribution, population distribution, and whether we should consider build the polyA tail into the plasmid or add it by the polyA polymerase. Those are options we can have. So polyA tail length directly impacts mRNA expression. So this is one study we had in-house that we compared different lengths of the ETLP mRNA with different lengths and then look at its expression. We can see from 40A to the 80A, increasing the polyA numbers will directly increase its expression efficiency. And for the polyA tail length analysis, which can be analyzed by the enzymatic digestion followed with small RNA biolyzer analysis method. So to achieve this, we cloned the polyA tail into plasmid, and then that which directly translate into the mRNA. We also compared the option of the building the polyA tail into a plasmid or added post-transcriptional with polyA polymerase. So the signature of the A peak here, you can see on the top right corner, that is a regular characteristics of the polyA tail added by polyA polymerase, and which will have a lower expression efficiency as analyzed. Third, next, maybe we can move on into the capping efficiency attributes for the mRNA. So for cap, there are options of uncapped. You can there is a cap 1, cap 0, or cap 2. So these are directly linked with the mRNA expression efficiency. So actually, for some plant-based mRNA or positive sense RNA virus, they like to use the cap 0 mechanism. And cap 1 is the most commonly used for the mammalian cells to they distinguish themselves and non-self mRNA using the viral infection. Cap 2 also impacts RNA protein production in a cell-specific manner. There's also a study showing that cap 2 mRNA can contribute to some immune evasion in the human cells. Next, we want to talk about the properties of chemical modification on the mRNA. So mRNA, we mainly studied 4 kinds of modifications: unmodified RNA, or the N1-methyl pseudo-U, or 5-methoxy-U, or the combination of 5-methyl-C plus pseudo-U. So here, there's a lot of data I'd like to share. So first, from the expression efficiency, N1-methyl pseudo-U and 5-methoxy-U modifications enhanced the mRNA expression efficiency significantly. And 5-methyl-C plus pseudo-U modification had a slightly weaker expression than other kinds of modifications, while the non-modified RNA has the lowest expression. We also observed significant toxicity from the non-modified RNA in this test. So from that, we also want to look at why the unmodified RNA caused a lot of toxicity, which we observed as a cell death in our study. So since the mRNA requires uridine modification to prevent activation of cellular innate immune sensors to reduce in protein expression. So these are the detailed studies look at the innate immune response of the different kind of chemical modified RNA in 2 cell lines, A549 or THP1. THP1 is an immune cell line that we studied. Look at the wild type U stimulated both the interferon and NF-kappa B pathways dramatically. And we also observed that NF-kappa B pathway activated much later than the interferon pathway. And the 5-methoxy-U reduced the least innate immune activation in our study. So this, the same result was observed in the another cell line, THP1 dual cell line. So we also analyzed the impact of this kind of chemical modification on this NF-kappa B and the interferon response pathway. Similarly, wild type U stimulated both interferon and NF-kappa B pathway dramatically, and the NF-kappa B pathway was activated later than the interferon pathway. And we observed the 5-methoxy-U modification caused the least NF-kappa B pathway activation. Yeah, same experiment we performed with a longer RNA because ESP-Cas9, which is 4.5 kb mRNA, because longer ones could have different performance. So we look at that and also observed that wild type U stimulated both interferon and NF-kappa B pathway dramatically, and NF-kappa B pathway activated later. So with that, we got the that conclusion from our end. We think U modification can enhance mRNA expression. So comparing intensity of the GIP signal, N1-methyl pseudo-U modification cause the highest, give the highest expression efficiency. However, the pseudo-U plus 5-methyl-C and 5-methoxy-U modification are similar. And wild-type U show the least EGIP intensity. The mRNA expression can be detected as early as 8 hours after transfection. Innate immune activation tightly related to the cell survival rate. The wild-type U triggered cell death starting from 8 hours after transfection, while the mRNA with modifications didn't trigger significant cell death. And we observed that 5-methoxy modification reduces innate immune response. So in both cell lines, the 5-methoxy modification showed overall the less innate immune activation and a good cell survival rate and a good protein expression efficiency. Yeah, at last we want to look at the impurities of the mRNA. There are multiple impurities that we analyze. Mainly want to discuss about 4 impurities: the plasmid DNA residue, the double-strand RNA residue, protein residue, and endotoxin level in the mRNA product. So plasmid DNA residue, why do we need to detect that? So those DNA residue can encode or harbor oncogenes or infectious disease, agents that lead to possible oncogenic or infectious infective agents. So WHO and FDA recommended that a 10 nanogram per dose of residual DNA from the reference we found. And we are detecting the DNA residue by the qPCR method. And this method can have a detection limit very low, and the product's residue here is less than 0.1%. double-strand RNA residue. So how is double-strand RNA generated? So it's kind of impurity that's generated during the in vitro transcription process by multiple ways, like self-complementary 3' extension or RNA-primed transcription or RNA-dependent RNA polymerase activity caused those byproducts of the double-strand RNA. So double-strand RNA has, triggers significant innate immune response. We developed the stop blot method is what usually used for this assay, and we further optimized to get into a linear range to detect about 0.25 or 4 nanogram of double-strand RNA per well, and we ensured the final product of double-strand RNA is less than 1%. Another thing we concern about is the protein residue. So protein residue, which is also another main impurities that from the enzymes used in the process. Generally, the guideline for the protein residue in biopharmaceutical products is less than 100 nanogram per milligram. So this nano-orange assay, I think, is widely used for the protein residue assay. We also use this one, and it has a very sensitive detection limit, 0.1 to 1 microgram per ml of protein in the sample, and we ensure the final protein residue in mRNA product is less than 0.1%. Endotoxin assay. Yeah, endotoxin is a concern when we use the mRNA in vitro or in vivo, mainly in vivo experiments. Endotoxin, generally you think less than 10 EU per ml of the mRNA product is good for the application, for multiple applications. There are multiple ways for this assay, either a semi-quantitative endotoxin assay by a gel clots kit or quantitative endotoxin assay. We used the kit from GenScripts that have developed with the semi-quantitative endotoxin that we can adjust the sample concentrations, make sure we can detect whether it's below or above 10 U per ml. To be more quantitatively, the quantitative endotoxin assay kit is good, can perform the job that can tell you the exact number of how much endotoxin are there in the final product. Yeah, so how did we achieve all this kind of the impurity residue control, which are performed by the purification process? And in the GenScript, we have the RUO grade mRNA and preclinical grade RNA. For RUO grade, there's a general column purification that can give you a very good results, or TFF method. And further, the oligo-DT-based purification can improve the integrity of the mRNA to a higher level. And actual double-strand RNA removal by the chromatography can significantly reduce double-strand RNA if it is generated in the IVT. But we generally optimize the IVT condition to ensure the double-strand RNA content to be below the required specifications. So this is a summary of the QC specifications that we talk about today, include the identification by different kind of properties, like polyA tail length, the mRNA length itself, the pH of the mRNA and the buffer, and there is also purity aspect attributes and the impurity attributes and the safety ones, include endotoxin and bioburden assays. So to summarize, the key quality attributes we talked about today include the plasmid DNA, which is a key starting material for the mRNA products. The quality of plasmid DNA is crucial to the final mRNA products. So codon optimization and polyA tail quality, supercoiled plasmid DNA ratio before linearization, and the purity of the plasmid DNA are key CQAs of the plasmid DNA for the mRNA product. Lastly, Next, for the mRNA integrity, which is significantly affected by the plasmid quality and IVT conditions, which will impact the mRNA expression efficiency significantly. So for polyA tail, the polyA tail length and the population distribution impacts mRNA expression efficiently. And the cap options that will also impact the expression efficiency, and it also differs by the cell type. The Cap2 mRNA can impact protein production in a cell-specific manner, and it can contribute to RNA immune evasion, as reported by a recent study. Chemical modifications on mRNA is required and important. So unmodified uridine will trigger cell death and reduce expression efficiency. And there are different kinds of chemical modifications can enhance expression and reduce its immune response. So the impurities we always want to control for the mRNA products include the residue of the plasmid DNA, the protein, double-stranded RNA residue, the endotoxin levels. Those will impact the mRNA expression efficiency and immunogenicity, toxicity. Yeah, with that, thank you very much for your listening. I would like to answer any questions.
Great. Thank you, Feng-Mei, for an excellent presentation. I see that we've received a few questions already in the chat, which I'll get to in a moment, but I will give everyone else a few moments now to enter any other questions they might have in the Q&A box to the left of the slides. I'd also like to, before we begin the Q&A, thank Asahi Kasei Bioprocess, Wuxi, Cytiva, Phenomenex, GenScript, and Polypeptide Group for sponsoring this Digital Week. Now back to Feng-Mei to begin the Q&A. So it looks like we have several questions in here, Feng-Mei. I'll start with this one. The intracellular immune response pathways are very interesting. Are there any extracellular immune response pathways to mRNA? For example, direct activation of immune cells?
That's a good question. So direct activation of the immune cells, I think that should require some interaction of the RNA with the immune cell receptors. Generally, like those double-stranded RNA, I think that will be sequence specific and structure specific. We haven't got a lot of data on that yet. I think there's a possibility.
Okay. The next one, how the specifications in terms of impurity how were the specifications in terms of impurity residue defined?
Yeah, actually, I looked into the references. So there's a lot of related study in the past for the biopharmaceutics. I think the mRNA shares a lot of common standard in the biopharmaceutics that from the FDA or WHO, like if it's an RNA product, like residue protein, residue DNA, those are shared standard. And there is a continuous study about how those numbers will affect the safety of the product. I read the literature. Along the way, there is a continuous discussion with the authorities, and there's amendments. Like I see from the plasmid DNA, those numbers actually we are very cautious from the beginning, and the standard got looser at the end recently. I think there is a continuous effort. So those numbers I reported, I just found from the references that's most up to date I can find.
Okay. The next question, so someone asking, what does RUO stand for?
Oh, that stands for research use only. So that grade of RNA that we only define that for the early phase development study.
Perfect. Next question. Did you run the AEX HPLC on your plasmid DNA template? And if so, what column did you use? Also, did you use AEX HPLC for mRNA integrity? And if so, what column?
Oh, okay. Yeah, those are for the specific method. For plasmid DNA, it's a AES plus size exclusion column from Shimaju. I don't remember the exact number, but I can search on that. And for the mRNA integrity, actually those figures I shared with you today, those are from Bionizer, not from HPLC.
Okay, great. And the next question we have, can you make mRNA at 10 kb in length?
Yes, actually for the longer mRNAs, it has more problem and degradation, so we got to optimize the ability condition further, but we do have the method that can have a high integrity of 10 kb mRNA.
Okay, great. And for the audience still listening, just a reminder, if you do have any questions, please enter them in the box. I only see 1 or 2 left, so I'll give you another moment if you have any to put in there. So the last 2 questions I see at the moment are, what storage buffer do you recommend for mRNA to preserve its stability?
Yeah, actually we performed the stability study. So far, for short RNA, it's okay, like water or the sodium citrate or T buffer, they are all stable. But for longer RNAs, definitely the sodium citrate buffer give you a better stability in long term.
Okay, perfect. This is everybody's last chance to enter any questions. Here's the last one I see. Does your company provide GMP-grade mRNA synthesis service now?
So at GenScript, I see the ProBio has ProBio subdivision of our company is providing the GMP-grade mRNA service right now.
Okay, perfect. Looks like that's all the questions that we have in the inbox. So with that, I'd like to thank you, Feng-Mei, for a great session. If anyone does submit a question or didn't submit a question that wasn't addressed, keep in mind that the speaker can reach out to you directly after the webinar. Also, just like to remind everybody that this session was recorded. You'll receive a notification in 24 hours when the on-demand session is available for viewing. Also, before you log off, please take a moment to complete the feedback form so we can continue to improve your Digital Week experience. And on behalf of Informa Connect Life Sciences, thank you all for attending and hope you all have a great day.
Thank you.